My name is Claire Tang. Today I am happy to present my paper, Discovering Unknown Diseases with Explainable Automated Medical Imaging. We all know that machine learning models are trained on massive history data from known diseases, and they learn to do automated diagnosis on the same known diseases. However, human knowledge about diseases are incomplete. New and unknown diseases, which we call out-of-domain diseases, always exist, for which data is not available at the model's training time. For example, COVID-19 was a new disease. Imagine you have a training dataset that consists of normal chest X-rays and the known pneumonia disease images, and we build a deep learning model with this dataset. When confronting an image of COVID-19, can our model detect that it is a new and unknown disease? Chances are it cannot. In fact, even doctors misdiagnosed COVID-19 to be a known pneumonia, such as SARS, MERS, in the beginning of the outbreak. This turns out to be a very challenging and open problem in the research community. In this project, I tried to explore how to enable machines to detect the emergence of new, unknown diseases when it is trained with only known disease data. In this work, we made two hypotheses. First, we hypothesized that existing models fail to detect the new, unknown out-of-domain diseases due to overconfidence in the model. Second, we hypothesized that careful calibration to mitigate overconfidence can enable deep learning models to do better in detecting out-of-domain diseases. First, what is true confidence? If a model has a confidence of 80% in each prediction, one would expect the model predicts 80 times correctly out of 100 predictions. But based on the paper in ICML 2017, it points out an empirical observation of ResNet 101 on CIFAR 100 dataset that there is a big gap between the model confidence and the actual output accuracy. Accuracy is much lower than confidence score. The pink region marks these gaps in the figure. I was intrigued by mathematical analysis and explanations on how this overconfidence happens and how we can overcome or mitigate overconfidence. Let D be a random variable. When X is in domain, D equals to 1. When X is out of domain, D equals to 0. Existing deep classifiers implicitly assume that all data are in domain, so what they actually model as, for an image X, and D always equals to 1, what is the probability of X belongs to an in-domain class? However, in open-world setting, we should model both in-domain and out-of-domain probabilities. Since unknown data is not available during training, for any image X, we can only train to model the in-domain class probability. When an image X happens to be out of domain, we will just implicitly deduce its out-of-domain probability. If we expand equation 2, we find that one of the items is actually pretty small. For example, for an unknown image X, Py in given D equals to 0 and X is very small because X is out of domain and Y in as an in-domain label. On the other hand, when X is an in-domain image, PD equals to 0 given X is close to 0. Keep in mind D equals to 0 means it is out of domain. Therefore, in both cases, equation 2 can be rewritten to be equation 4. Let FCX be the logit for PY in given X, and FDX be the logit for PD equals to 1 given X. After softmax normalization, equation 4 becomes equation 6. Here is a simple example of equation 6 when applies to an unknown image X like COVID-19 to a two-class classifier. In the figure, the x-axis shows logits before normalization and the y-axis shows their probability distribution after softmax normalization. Assume the logit FCX as 0.5 and 0.8 respectively for the two classes, normal versus pneumonia, pretty close numbers. See blue points in the figure. With FCX divided by FDX, these two numbers become 5 and 8. See the red points on the x-axis. After softmax normalization, 5 and 8 becomes 5% and 95%. This is exactly how overconfidence happens. The model makes wrong classification but with very high confidence. Based on the mathematical analysis, I show a simple scaling approach to mitigate the overconfidence. For example, very simply, compensate overconfidence with a large number T. Refer to equation 8. With that, we mitigate the overconfidence, and the normalized probability goes back from 5% and 95% to more reasonable numbers like 43% and 57%. With these numbers, our model cannot confidently conclude it is either normal or a known pneumonia image. Therefore, it is an unknown. Based on the mathematical analysis and explanation, we show another approach that completely removes the softmax normalization layer to avoid overconfidence. We introduce a new layer named generative classifier layer. It learns a Gaussian distribution for each class. With our new confidence calibration methods, we built a new deep learning pipeline in which our new method will enable better detection of unknown diseases. During the training phase, we not only learned the weights and parameters for deep classifier, we also learn the parameters for our new confidence calibration function. In testing phase, when we see an image, our system will first do inference and then calculate confidence using our new calibration method. We then decide if it belongs to a known type of diseases or is an unknown. If it belongs to a known type, our normal deep classifier will classify it. If not, our system will conclude it is an unknown. We also designed a novel approach to give visual evidences on the suspected regions that lead to the decision of unknown. This can help doctors can look at the visual evidences and make the final decision. When the project started last year, I was focusing on a skin dataset. I had a big dataset from ISIC, International Skin Image Collaboration, website. I participated in their challenge for lesion classification. It contains a dataset of unknown skin diseases. Later, when the coronavirus broke out, I downloaded COVID-19 chest X-ray images from various websites. These images contains doctor clinical notes. I trained deep classifier using the known disease datasets and tested on the unknown disease dataset, for example, trained on known pneumonia dataset and tested on COVID-19 dataset. More specifically, we trained several ResNet-based classifiers. ResNet 152 performs the best. We used some well-known metrics in literature to measure our model performance quantitatively. As you can see, our new calibration methods significantly outperform the baseline methods. Here are some implementation details. We implement our code using the PyTorch 1.1.0 framework. The experiment is run on 8 NVIDIA GPUs, Tesla V116GB GPU. Training images are normalized using mean and standard deviation. When preprocessing data in the PyTorch data loader, we resize each image to be 256 × 256 and then randomly crop each image to 224 × 224. We also augment the training images with horizontal and vertical flips as well as zooming. We train several ResNet models, including ResNet 50, ResNet 101, ResNet 152. We set learning rate to be 0.0001. We all know that deep learning is a black box. It is hard to know what happens inside the black box. To help doctors understand why our model makes that unknown decision, we also designed a novel visual explanation method to highlight the suspected regions when our model makes the unknown data detection decision. We backpropagate its gradients into the CNN players. Using the weight for each pixel based on its corresponding neuron importance, we generate a heat map M. Note, in a normal visual explanation for in-domain classifier, the inside sum term marked in green as not needed. M consists of a grid of numbers, different numbers in M map to different colors in visualization, indicating the different importance of the pixel region in the decision making. For example, a high umber means it is important to the decision making, we mark it as red. A small number indicates it is not important, we mark it a green. Next, we show some example results from our visual explanation of unknown disease detection when we tested on skin lesion and COVID-19. In the first example, we show the baseline model made a wrong decision and yielded a heat map indicating that it looks at wrong regions. Keep in mind, we backpropagate the gradients about the wrong decision to the CNN layers to generate this wrong heat map. In contrast, our heat map, the important suspected region, is quite consistent with doctor clinical notes, right upper, left lower. The visualization also shows that our model looks at the skin lesion region very similarly to the ground truth region that dermatologists labeled. In the second example of our result, baseline model looks at two broad regions, leading to a wrong decision. In contrast, our model looks at much more focused regions. The third example shows the baseline models look at two narrow regions, leading to wrong decision. The fourth example shows again the baseline models look at wrong region. And our model does not find evidence linking to the known disease, thus concludes it is an unknown disease. As conclusion, in this project, we mathematically analyzed and explained why existing models fail with overconfidence, proposed new calibration methods to overcome overconfidence. We validated our hypothesis by quantitative experiments showing significant performance improvement over baseline using two different types of datasets, skin lesion and chest X-ray. Using just-in-time real-world COVID-19 data, we tested our novel method and successfully detect the new COVID-19 disease with a deep learning model trained only with the known pneumonia image data. Our novel visual explanation method are also consistent with doctor ground truth labeling and clinical notes. We will continue to validate our methods with more datasets and also larger datasets. With COVID-19 image data increasingly released, we are interested in working on machine learning models that can better understand COVID-19 and also predictive models based on such understanding. In particular, we are interested in few-shot learning that can quickly learn new models based on small amount of new data and also multimodal machine learning models that can leverage other modalities like text. Thank you for listening.